Hey Beaklings, I said we'd try those 5 practice problems together. Okay, so let's give them a whirl and see how you did. Okay, pause the video if you need to make sure you're solving them correctly. Okay, so we're back into this worksheet that we started yesterday, Introduction to Genetics, and hopefully this made sense as we solve the problems together that were in here. So I asked you to try these 5 practice problems. So here we go. Number 1 says, imagine you are Gregor Mendel. Start an experiment by crossing these parents, a true breeding tall pea plant and a short pea plant. Get the phenotypic and genotypic ratios of the F2. Show all Punnett squares. All right, here we go. Okay, so for problem number 1, we're on, right? We have tall times short. Now the problem told us well, all we had to know to start, right? It said we have a true breeding tall plant. And what that says to me, well, first of all, tall is dominant. We know that. So if you're a true breeder tall, you know that you're going to be big T, right? And short is going to be little t, little t. You have no choice except to be a homozygote if you're short, right? So this is definitely a true breeder for that gene. Okay, for that allele. So let's get our F1, and you know how we do this. We have our big T, our little t, and this is going to be our F1 generation possibilities inside of here. And no matter how you do it here, you can see that you're going to get all big T, little t, big T, little t, big T, little t. So our F1 is going to be 100% tall. Big T, little t. And Mendel was not surprised by this eventually, right? He said, yeah, it's not a blend. The short is in there somewhere. But this is not what the question asked us to do. It asked us to get the phenotypic and genotypic ratios of the F2, right? So now what did we do? We take these plants, and again, these are possible plants. They're not actual, but we take 2 F1 plants, and we cross them to themselves. So we do big T, little t times big T, little t, and now this is going to get us our F2. Okay, so we have a heterozygote. We're going to cross that to a heterozygote. Okay, so we had a monohybrid cross, and now we're going to cross these together down here. And we're going to see that we get, well, let's take a look at what they would look like. Okay, and the expression of these genes, okay, that's your phenotype. So the phenotypic ratio is going to be what? Well, I see tall, 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 short. 3-to-1 tall to short. That's what I see. Now the genotypic ratio, that's a little different because that's what the genes are. So in this case I have homozygous dominant. I have 2 copies of the dominant allele. I have 1 big T, and I have 2 heterozygotes, 2 big T, little t, and 1 little t. So there's your answer for the F2 generation. We have a phenotypic ratio of 3-to-1. And a genotypic ratio of 1 to 2 to 1. All right, so now for practice problem number 2, it says cross a heterozygous yellow-seeded pea plant to a green-seeded pea plant. Show the Punnett square and describe the possible offspring. So again, they're telling us everything we need to know, what's dominant, what's recessive, excuse me, because it says heterozygous yellow. So that would mean that, oh, it's yellow, but it has 2 different copies, so the yellow must be dominant, right? Whereas green, they don't give us any more information, so green must be recessive. Okay, hopefully that makes sense to you at this point. So let's go ahead and solve number 2 together. All right, here we go. So it said number 2, a heterozygous yellow. Now I would have to look back at my sheet if I didn't know this, but I would find out that yellow is dominant over green, right? So my symbols again, big Y, and let's make little y for green here. So a heterozygous yellow plant would be big Y, little y, and you cross that to a green plant would be little y, right? That's all. So if you did your Punnett square correctly, that looks like a G, that's supposed to be a Y. I would have yellow, green. So in this case, my phenotypic and my genotypic are 50-50, or what you could say is 2-to-2, or what you could say is 1-to-1, right? These all mean the same thing. It means that 50% are going to be yellow, 50% are going to be green. Okay, not too bad, I hope. Let's move on to problem number 3. And it says, imagine that tall and short pea plants were observed growing in Mendel's garden. These plants were identically grown under strict environmental conditions. For which phenotype would you know the genotype? Well, this problem is simply saying, if we go back again to number 1, it's just talking about tall and short, and we have already discovered that tall is dominant to short. So all this question is saying is, hey, which phenotype do you automatically know the genotype? Right? So for number 3, we know the genotype of the what? What do you think? Dominant or recessive? Right? The recessive. Okay, so in this case, short, right? Because you know short has to be little t. But if we were tall, oh, that's right, because we could be big T, little t, or homozygous, big T. Okay, so that's all. It's just trying to make sure you understand that you would know the phenotype of the recessive. All right, let's move ahead here to number 4. Okay, so let's try working with 2 genes at once. All right, so this is one of those bigger squares, right? Where you have to FOIL and put the 2 genes or 2 alleles on top and to the left. Like Mendel did when he discovered independent assortment. What is the phenotypic ratio of the following cross? So a short heterozygous purple pea plant, and if you're good, you see short, oh, that must be little t. Heterozygous purple, if you're heterozygous, purple must be dominant, so big P, little p. Heterozygous tall, so the opposite here, same gene, right, different alleles, big T, little t. Heterozygous purple, big P, little p. All right, so this is going to be a little bit different than what we did yesterday. So let's give that one a try. Okay, so here you can see problem 4. We have a short heterozygous purple to a heterozygous tall heterozygous purple plant, right? And as you can see, there's 2 genes, and how we start, we wouldn't even be able to put these symbols unless we knew what was dominant and what was recessive. So obviously we're going to need a 16 square Punnett square, right? And then we're going to go ahead and FOIL and see what we end up with. All right. Wow, that's not too even, huh? So here we go. We are going to FOIL or distribute our first T and then our second T with our P's here. So the first T with the first P, and now my outer T with my outer P. And technically we could stop there. I'll show you what I mean. So now it's my inner T with my inner P. That's the same as this. Okay, and then my last T with my last P, that's going to be the same as this. So technically we could just ignore these last 2 columns, right? Or you could do all 16 and then just cut it in half, right? Reduce it. But anyway, let's do the other parent. So now let's put this parent down the other side and FOIL that out. So my first T with my first P, right? And then my outer, inner, and last. Okay, so now just go ahead and fill in your Punnett square. You keep your capital first, right? And keep your genes separated. So this would be big, little, right? This would be big, little. Okay, and then this one would be big, little. Over here we have little, big. Here we have little, big, little, big, little, and little. Okay. And again, you could do these 2 columns as well. It's just, you know, this block is going to be the same as this block is going to be the same as this block, you know, this one. So anyway, I'm just going to fill out these 2, right? Because I already know it's just reduced for me. All right, now, if you remember how I like to solve these is, I like to think of what we could get. So if we crossed a short purple, right, to a tall purple, obviously we can get the dominant, right? So the question asks for the phenotypic ratio. So I don't really care about the genes right now. I'm just looking at the genotypes. I'm just looking at the phenotypic ratio. So for tall, I know I need a big T. For purple, I know I need a big P, right? And then I could also maybe get a tall short. Oh, what am I doing? Tall short. There it is. Mistake on camera. Ridiculous. I could get a tall white. Right? I'll even mark that on the board. Right? I mark my mistakes on the board, and that'll be number 2, I think, for my life, maybe. So tall, and now white would be little p. Okay, and then, excuse me, we could also maybe get a short purple. And you know how to get a short purple. It'd have to be little, big P something. And then maybe we get a short white, completely recessive. And that'd be little. Okay. So now we go back in and we just take a look at how many did we get? All right, so tall purple. Let's see here. I count tall purple, tall purple, tall purple. Okay. I counted 3 of those. Okay. Now, it could be a 6 if I did the entire Punnett square, but I'm just going to consider these 2 rows, and I'll explain that at the end. Now let's look for tall white. Well, I see 1, right? So let me go back to blue here. I'll circle it with blue, and I see 1 of those. All right. And now let's take a look for short purple. Yep. So I'll put a little star. 1, 2, 3. All right, we got 3 of those. And now the dark black short white. Yeah, right there. Okay. So technically what we got was a 3 out of 8 to a 3 out of 8 to a 1 out of 8 to a 1 out of 8. Or what you could say is a 3 to 3 to 1 to 1. Right? That's our ratio for that cross. And if you did all 16 squares, yeah, sure, you could count it that way. That would just be a what? That would be a 6 out of 16 to a 6 out of 16, right? And now you'd have a 2 out of 16 to a 2 out of 16, right? Or a 6 to 6 to 2 to 2. Either of these is correct, right? They're both the same answer. All right, because it's all based on what are your chances? What are the odds of getting any of these potential kids in there? All right, so that one's a little trickier. That's a 2-gene cross, and you see that we got a little bit of a goofy ratio here. In the packet yesterday when we saw the dihybrid, if that was a big T right there, so big T, big P, little p times big, little, we would have gotten a 9-3-3-1 ratio. But see, because this is homozygous recessive, that changed everything, and now we get a 3 to 3 to 1 to 1 ratio. All right, and let's take a look at number 5. Okay, so number 5 says, if you cross true breeding smooth seeded pea plants to wrinkled seeded pea plants, the F1 will be 100% smooth. Explain why the recessive phenotype disappears in the F1 generation. All right, so easy enough, right? It's just because it's recessive, but let's take a look. All right, so I probably should have, I call it smooth, but Gregor Mendel referred to it as round. So to start this problem, maybe I should just say when you see a smooth pea, that's basically the same thing as saying a round. Remember yesterday we did the round yellow times the wrinkled green. So smooth or round is dominant over a wrinkled pea. Okay, so I'll just change it up here when we talk about it. It said a true breeding smooth or round in this case, so I could write, you know, smooth or round, same thing. True breeding. Okay, let's stick with R's like Mendel did. So we have a round times a wrinkled is what the problem said. So little r. Oh, well, yeah, of course. In the F1, you're going to get 100% that, right? And if you don't know how we got that, simple little Punnett square, right? Okay, so it says, why does the wrinkled completely disappear in the F1 down here? Okay, so this is the possible F1 kids. Well, you know that it disappeared phenotypically, but not genotypically. You know that that recessive is in there, and the only way for a recessive to show up is if there's no dominant allele around, right? Because dominant alleles are expressed no matter what if they're present, whereas recessive alleles, they can't have any dominant around. All right, so that was the end of that, problem 5. Hopefully that helped. Okay, so I hope that was helpful to you. Again, if that is still confusing, it's all right. We're still pretty new into genetics, okay, but it will get easier as we go along. Feel free to shoot me an email if you need to. Okay, take care.